Hi, we're here at the 89th Annual Symposium at Cold Spring Harbor, and I'm joined by Professor Navdeep Chandel from University of Northwestern in Chicago. Nav, thank you so much for joining. So your lab studies mitochondrial function in various physiological contexts. So there's a lot of models that link mitochondrial function or dysfunction to aging. And considering the many facets of mitochondrial biology and the many roles they have in cells, how do you approach mitochondrial roles in aging?
Well, it's nice to be here, and that's a great question. It's the key question, right? And there's always been a link between so-called mitochondrial dysfunction and aging. In fact, every version of the hallmarks going back 15 years, one of the staples continues to be mitochondrial dysfunction, right? It's one of the original hallmarks, and as they've broadened the hallmarks, there's more. In some ways, if you look at it from a historical perspective, it's arguably one of the oldest theories of why we age, right? And going back to the 50s, or where people proposed the mitochondrial theory of aging. And in other words, that as we age, our mitochondria are dysfunctional. Maybe their mitochondrial DNA even picks up mutations. And over time, certain tissues just degenerate because you just don't have enough mitochondrial function. Okay, so what is that mitochondrial function? Well, the textbooks tell you it's all about ATP. It's the bioenergetics paradigm. So it's such a beautiful idea if you think about it. If you're at a cocktail party, I can explain this in a heartbeat, right? What do mitochondria do? They're the powerhouse of the cell. Great. Okay, so the powerhouse of the cell gives you ATP. Okay, you can understand that. Everyone can. And what happens during aging? The powerhouse just, the engine isn't working so well, and before you know it, you run out of gas. You run out of the energy in a particular tissue. And therefore, that tissue degenerates. Maybe you get dementia, maybe you don't have enough lung function, definitely the muscles. So, I play soccer, I love to play soccer. I can tell you, every 10 years there's a step down function. Why? I don't understand it. Some people would say maybe your mitochondria are not as rejuvenated as they were when I was a younger man. And so this is the bioenergetic paradigm. And to be honest with you, it can never be discounted, because how do you give ATP back? It's almost untestable in some ways, right? Because if the idea is your mitochondria aren't making enough ATP until 90 or whatever age you live, how do you give back ATP, right? I can't inject ATP, it can't get into a cell. Well, so that's a difficult theory to test. Of course, the other one was equally old, is that mitochondria get dysfunctional and they make lots of ROS, oxidative stress. So you should take dietary antioxidants, and there's plenty of them, vitamin E, vitamin C, and that will help you live longer or healthier. Well, dietary antioxidants so far have failed largely, right? And one of the reasons is, I would argue, like in some cancer studies, it make the cancer worse, right? In health span assays in humans, it hasn't really looked like a winner. Now, you could argue we don't have the perfect antioxidant, right? Okay. But the other thing is a recognition, and this is something my lab has really worked on, and I've been working on this since a graduate student, that it's not as simple as mitochondrial oxidative stress bad. It's that mitochondria make H2O2, which is a signal, and that signal is beneficial. Now, other people years later showed that H2O2 from mitochondria can eventually become a lipid radical and cause ferroptosis, which is a form of cell death. Right? So you can imagine, as a tissue is degenerating, maybe you need an antioxidant that prevents ferroptosis. Maybe. Or the other thing is, maybe there's enough antioxidant capacity all the time we have all balanced just to give you that H2O2 signaling. And we found, for example, like in a recent study, that the mitochondria making H2O2 is absolutely necessary for proper IL-10 release. IL-10 is the major anti-inflammatory cytokine. If you lose your ability to release IL-10, you can get colitis, you can get a lot of autoimmunity, right? And so there's a beneficial effect of mitochondria as a signaling organelle making H2O2. And so I think the big shift in the last 25 years has been the recognition beyond ATP that mitochondria do a few things now. So I just told you about H2O2. Their genome can get released. the DNA and mitochondrial DNA and RNA, and it can activate inflammation like C-gas sting pathways or the MAVs, RIG-I MAVs pathways is to turn on inflammation, right? All the epigenetic enzymes, you could argue, are run by mitochondria. So acetylation, where do you get your acetyl-CoA? The citrate, and in large part from mitochondria. Many of the demethylase reactions for DNA, RNA, and histone, they're alpha-ketoglutarate dependent. Alpha-ketoglutarate is a TCA cycle metabolite. So you can see how the control of TCA cycle metabolites is now impinging on the epigenetic landscape. The integrated stress response has a mitochondrial input into it, right? If mitochondria ATP even goes down, the cell responds by activating AMPK. And if you activate AMPK, you activate autophagy, you shut down anabolic processes. So I would argue one of the excitements in the field is mitochondria are signaling organelles. They're communicating for inflammation, epigenetics, controlling catabolism, anabolism, integrated stress response. I think one of the big questions is how does it happen, right? How does these things get released? What are their biological targets? And can you manipulate just the signal from mitochondria and show that it's having a dominant effect in physiology? But let's assume it is. So one could argue maybe with mitochondrial dysfunction during age, if there is any, is what's happening is something like maybe too much mitochondrial DNA is being released. Mm-hmm. Or you're not activating enough H2O2. Not too much H2O2, too little H2O2. All right, you're losing a physiologic response. So I think we think of mitochondria in that angle. Now, what I would say is some interesting experiments that have been done trying to test what mitochondria respiratory function is doing. So we did a simple experiment where we took a catalytic subunit of mitochondrial complex 1. We knocked it out. So the electron transport chain activity goes down by 50%. So you and I have 100%. Now you're at 50% in development. And we just aged the mice. Nothing happened. It's a completely negative study. It's the most expensive study we've ever done, right? Viral infection, totally fine. Obesity, totally fine. Rotor rod, open field, dementia, whatever you want to do. Yeah. Every test, totally fine. And why is that study important? And I think other people have come to this conclusion. You and I are born with excess mitochondria function. So you're born with, let's say, 100%. What the primary mitochondrial disease people have taught us is, so those are mutations in a mitochondrial protein, and you have to get at least 70 to 80% decrease in that function of electron transport chain function, respiratory function. And then you get a pathology in a certain organ. When I've looked at the data, you can see a 40% decrease in electron transport chain activity, maybe 50. Yeah. You rarely see 80%, right? That's sufficient to trigger. So, I'm not completely convinced we're running out of enough gas, right? The analogy is I take the bus to work still, but I'm not a good driver. Everybody knows that, it's fine. If you have a nice, beautiful new Ferrari, goes 400 kilometers, mm-hmm. or let's say in 100 years, now it's 200 kilometers. In the city of Chicago, you can never go over 100, or you get a speeding ticket. So you're not rate limited. Even though you have a 50% decrease, that's enough to get by, right? Maybe you're not a race car driver, but it's enough to get you around town. Mm-hmm. So I think mitochondria is the same way. The respiratory function might decline, but it's not rate limiting. And what might be rate limiting, or what might change, is maybe something else isn't being made. Alpha-ketoglutarate levels maybe decline, or the integrated stress response is always active, or not enough H2O2. And that's something we're sort of testing.
Right, and it's really interesting to try to tackle physiologically which homeostatic levels of which mitochondrial functions are critical. And one drug that has been suggested to target mitochondria in various contexts is Metformin, which right. you've also recently studied.
Right.
What did you find?
Yeah. So I have to tell you, it was an interesting story how I got into Metformin. It wasn't Metformin before the GLP-1 revolution, agonist revolution. It was the first line therapy for diabetes, and I think hundreds of millions of people have taken right. it. I got into it from the cancer angle. So there was always these prospective studies, retrospective studies, where they looked at all these epidemiological studies, and there was always a signal. People who took metformin, obviously for their diabetes, had a little bit of a lower incidence of a variety of cancers. There's an even cardiovascular events, etc. You could argue maybe they got healthier because of the diabetes, but even when they try to control for that, there was always a signal. So people started to think, well, let's try it in mouse models. And they did. And you could see in some mouse models, metformin had a pretty robust effect in decreasing tumor growth. Okay, so then people rushed right away to do clinical trials, eventually resulting in a phase 3 clinical trial in breast cancer that failed. And then that sort of, I think, in part, closed the book on metformin and cancer. Now, how did I get into this? I got into this because Reuben Shaw, well known for his AMPK work, which people thought was the main target of metformin. We can talk about that in a second. He told me, this is the joy of coming to a meeting, okay? You can't do this over, one mantra I have, you can't do this over Zoom, etc. I was watching a talk and he said, you know, Nav, a lot of people think metformin inhibits mitochondrial complex 1. There's all this data in vitro, in cell culture, it clearly decreases respiration. And I was like, well, what's the evidence for that in vivo? And so I started thinking about it, and I remember there was a great paper in the mid to late 90s from Takoyagi's lab, beautiful little paper. And he showed that if you took a yeast protein, one protein, it forms a dimer, it kind of functions like a mammalian mitochondrial complex 1. So he would take a cancer cell, give it like rotenone, a classic complex 1 inhibitor, oxygen consumption goes down. If he overexpresses this yeast protein, it comes right back up. And what it did is similar to complex 1, it would regenerate NAD, it would pass 2 electrons to the rest of the electron transport chain. It wouldn't proton pump, which is part of the ATP, but the other complexes would proton pump. So it would just restore respiration and ATP synthesis. It was a great rescue or restoration of complex 1 inhibition. So I said, well, why don't we go and take these models that people use where they show metformin decreases tumor growth. Let's put in this yeast NDI1 and see if it restores. So bingo, that's what we got. Metformin decreased tumor growth. You put the NDI1 protein in those cancer cells, tumors went right back up. And that was pretty good. This was a paper we published in eLife almost 12, 13 years ago. I will say, since you're an eminent editor, many of your colleagues, especially in the little journal that comes out of London, reviewed it but passed on it. And part of it is, I understand it, because everybody thinks they figured out metformin. It's very controversial. We put it in eLife, and I'm happy to report more than 1,400 citations. And then people started to really look to see if metformin, at least in cancer, works by inhibiting complex 1. So one nice study from Jason Locasal and Ernst Lengel was they took patients for ovarian cancer and they took biopsies and they saw TCA cycle metabolites were down and metformin accumulated in those tumors. So really started to suggest maybe that's how it works. But the clinical trials eventually failed. Yeah. And our own data showed that if we took, the NCI 60 panel cancer cells and we did metformin, some were exquisitely sensitive and others were not. So why the heterogeneity? And same in patients, I would argue. Yeah. And so we did a CRISPR screen and we asked a very sensitive cell line, and what protein, if you lost it, what gene, if you lose it, makes you completely refractory? Top hit, the plasma membrane transporter metformin.
That makes sense.
That makes sense, but it tells you something very insightful. Yeah. That if you're going to do a clinical trial, it's like HER2 and Herceptin, right? Yeah. You would never give Herceptin without HER2 positivity, I think. I would redo a trial based on whether the transporters are there. Yeah. If it's there, maybe you got a chance of giving metformin. If it's not there, it doesn't work. Right? But nevertheless, if there's enough transporters, metformin's anti-neoplastic effects are by inhibiting complex 1. And it's probably because it maybe activates AMPK as a downstream, increases NADH, which will decrease aspartate, the metabolite for growth. There could be many reasons why it's doing that. Probably the best clinical data as an anti-neoplastic agent comes from 2 papers in Nature and one in Nature Communications that just came. These 3 papers just came out, and they showed that people. One of the papers showed in the UK Biobank, if you took metformin, even if they controlled for diabetes, etc., that you had less clonal hematopoiesis. Now, as you know, clonal hematopoiesis is linked to cardiovascular disease, cancer, etc. But people on metformin had lower incidence of clonal hematopoiesis. In the UK Biobank, there are mouse models of clonal hematopoiesis. It decreased that, given metformin in mice. And then they did my NDI1 trick in one of the papers, which is great.
Oh, that's very nice.
Yeah, they could rescue the effects with NDI1 expression. So it really suggests that metformin preventing these clonal hematopoiesis, these clonal cells that outgrow and compete out wild type cells because they have some driver mutations, and that advantage is decreased if you have metformin. And so I think, to me, it's very satisfying, something we did almost 12 years ago and other people are seeing in different contexts. So, so that's its effects on cancer.
Right.
But as you know, the big effect, of course, why it's used is for this diabetes effect. So we've been working on that. So what we did was Colleen Rezik in my lab generated a mouse where she put NDI1 in the ROSA locus, and you can conditionally activate NDI1 in any tissue you want. So now a tissue will have the endogenous complex 1, and right next to it it'll have metformin. It'll have NDI1. So if you give metformin, presumably this doesn't work and this takes over in a given tissue, right? So she generated a whole body NDI1, and she found that the glucose-lowering effects, it's not fully, but largely were abrogated if NDI1 was there, suggesting that mitochondrial complex 1 might be the target. Zach Zebo followed this up. One of the things we did is we looked at clinical data, naturally. Where does metformin go? So you take it orally. You have to take it orally. First pass is the gut. So the intestine is where you have almost a millimolar. By the time it leaves in the portal vein, it may be 50, 70 micromolar, maybe even less in the liver, and then it goes to the kidney, and then you sort of pee it out, right, the bladder. But the highest concentration is in the gut. And we know that typically you need above 100 micromolar to inhibit mitochondrial complex 1. So we thought maybe we should do the gut experiment. So we did a Villain-Cree, put NDI1 in, activated in the gut. And what Zach found was largely when he gave metformin, again, the glucose-lowering effect, if he just put the NDI1 in the gut, it brought it, not completely, but it brought it back up. Again, suggesting complex 1 in the gut. Yeah. If you do an FDG PET, right, like if I took metformin and did an FDG PET, my gut would light up. In fact, patients who go in for FDG PET and have tumors are told to get off the metformin because it interferes. Right, so we did the FDG PET. It goes up with metformin. If the gut has NDI1, it doesn't go up.
That's really
right? So it suggests that. Now, it's not a full restoration or a rescue, and I think partly because I don't think the gluconeogenesis effects of metformin, lowering the liver ability to make glucose, I don't think that's going to be complex 1. I don't know that, but just because I don't think the concentration gets high enough. So there's likely another target, and I'm totally open to that. The other thing is, because we have both of them competing, the endogenous NDI1, we're doing the experiment, we're getting rid of the endogenous and only have the NDI1 in the gut to really, see how much of it is all complex 1. My guess is largely gut complex 1, but there's likely to be a component that's the liver, and that's probably another target of metformin, and I don't know what that is.
But so do you think that metformin actually directly targets complex 1 in the gut?
Complex.
Or
it's direct targeting of complex 1, yeah.
But there have been no structural studies or...
There was a nice structural study, a pretty nice one from Judy Hurst, published in Science. She had to modify the biguanides, which is the class of compounds like metformin, and then she mapped out a couple of sites. We've done where we've taken an isolated complex 1 and looked at NADH oxidation, and we can inhibit it. We've done isolated mitochondria. I think it likely binds to somewhere in complex 1, and if NDI1 is there, it says go ahead and bind, I'm good to go. So two effects I can explain somewhat by complex. One, the anti-neoplastic effects, as well as the lowering of glucose. So then people always ask me, well, why don't you take it? Well, I exercise, I have a reasonable diet, I get sleep. The only reason I could think of is maybe as an anti-inflammatory agent. So it has a good effect on the microbiome, and there's always been some link as an anti-inflammatory agent. So the best one comes from the COVID study. So they put a bunch of people within the first 3 days of metformin, if they were SARS-CoV-2 positive. They also did ivermectin and one of the serotonin uptake inhibitors, I think, like Prozac. Don't quote me on that. Memory's a little jot. But metformin had a signal for long COVID. It decreased. And COVID, we can argue what it is, but it is a disease driven by inflammation, especially the long COVID. And it lowered, it reduced some of the symptoms. And there's always been a link. So we did a study years ago with Scott Buttinger and Gokhan Mutlu. I mean, really, they led it, where they gave Beijing or Dusseldorf or Mexico City level pollution to mice continuously. These mice get a spike in IL-6, and they're more prone to cardiovascular events, exactly what humans are, right? But if you gave metformin, IL-6 was down, and their thrombosis was down. And so luckily, the city of Chicago is not as polluted anymore. It's actually one of the least polluted cities in the world. But I'm not joking when I say this. If I was in a polluted city, Delhi, a city I know well, I'd be on metformin for that reason more than any. Maybe, should everybody else it because of the anti-inflammatory? I don't know. I think one of the issues is people have focused on cancer and diabetes. Is in general overall health span assays. I would love to do work in the clinical setting of putting people, healthy people, middle-aged folks like me, on metformin and measuring CRP or other measures of inflammation over a course of a year or whatever. I don't know how you would even design the study to get an answer whether it's a good anti-inflammatory agent or not.
That's really interesting. So you think that in the aging sphere or in the health span control, it's more about inflammaging that we need to look at, and less perhaps metabolism and classical metformin mitochondrial studies.
Yeah. So one of the studies that got a lot of interesting play was a study in Cell about epigenetic clocks and monkeys, male monkeys, given us 6 years. I think they were given metformin, and they did all sorts of omics on them, and they found their brain function was better, and they showed that the inflammation was down. And so I would argue, especially in the healthy, right? I have a saying, the slightly metabolically unhealthy will all take GLP-1. The healthy, perhaps, whose weight is fine and they exercise and sleep, etc., and have a good diet, and maybe they might take metformin for the anti-inflammation, because they don't want to take NSAIDs, right, which are a little bit more potent. And maybe the microbiome being a little positive, maybe. But someone's got to do this properly, clinically,
Yeah, that's exciting. We'll have to see what others do in this area, but this is really exciting, and lots of exciting directions.
Yeah. So this is, I think, trying to figure out metformin's inflammation, right? And aging sort of is the buzzword that explains everything about as we age, the health span of age, and whether metformin is lowering that to improve that along with diet and exercise, etc. One thing that Zach did find in our group, which I think was quite clever, there was two things he found I think that would interest people. One was, instead of doing a classical glucose tolerance sort of test, you fast, what he did, he fast the animals and he just let them eat, right? Which is what you and I would do in the morning. So you get a postprandial spike in glucose. We all do. Yeah. And there's always been this link that if the higher the spike of glucose than an insulin spike, that's bad. Well, what he found, if you took metformin and then you ate, you had less of a spike.
Oh, interesting.
Right? Because what's happening is the intestine is sucking that glucose in and metabolizing it to lactate and then excreting it out. And he found less insulin spike.
Which is the goal.
Yeah, which is the goal. And of course, NDI1 rescued all of that.
Oh, very nice.
Right? And GDF15, which can get released, and LAC-Phe, which people have shown as sort of other good things that the gut releases, all of those are abrogated by having NDI1. And so I think there's something interesting to think about in those contexts.
Yeah, this is all very interesting. Thank you so much for sharing and,
Yeah.
and for chatting today.
Yeah,
This
it's been
has
great.
really great.
Yeah, thank you for your time, and you know, we'll continue working on metformin, and maybe get, hopefully with our insights, gets clinicians to relook at it from different aspects.
Yeah. I look forward to hearing more
Great.
in due course.
Thank you.
Thank you.